On March 24th of 2020, I was standing in the anteroom of a biosafety level 2 tissue culture facility over on Emory's campus in the Whitehead building. I was standing there with 7 or 8 colleagues because in the past months leading up to that March date, COVID had clearly started moving across the ocean. It had found its way into Seattle, and it was in cruise ships, obviously, across the globe. And there was increasing understanding that it was not going to be contained, as the Trump administration at the time was telling us that it was going to be. It had found its way into a number of different cities across the U.S., including in Atlanta. Emory University had decided to ramp down operations, in-person classes, things like this at this point, basically due to the deaths that were coming off the West Coast. So we sat in that room and we talked with a biosafety officer about the possibility of being able to study the blood of patients that were in the ICU in Emory hospitals that were dying of COVID-19. At the time, if you remember, there weren't many masks. There were no N95s. Hospitals were struggling to find the personal protective equipment, the PPE that they needed. And so while normally a pathogen like SARS-CoV-2 would be treated as a biosafety level 3 pathogen, where we would have disposable gloves and we would have respirators and all of those sorts of things, it simply wasn't going to happen. So we didn't have N95s. We had some KN95s later, but at the time, really what we got was a face mask and a face shield, those splatter shields, to prevent liquids from getting into your nose and your mouth. Nobody understood whether SARS-CoV-2 could get into the blood of a patient that was dying of COVID-19 and be reasonably passed from that patient to somebody that inhaled droplets. So we were advised when working with the blood that we should try not to make any aerosols, try not to aerosolize the blood that we were working with. And that we would double glove. So we had 2 pairs of gloves rather than the elbow length gloves. We had the little lab gloves, and we had some disposable gowns that we had managed to source. So anyway, we had decided to work on SARS-CoV-2 because we knew that it was pressing. We had some expertise. I was working in Yaki Sands' lab at the time. We were very good at understanding B-cell responses, and those are the responses that are involved in antibody production. We understood how to look at those responses in the context of sort of broken immune systems, or immune systems that have targeted their own tissues. Right, so we're talking about autoimmune diseases like lupus and rheumatoid arthritis, Sjogren's syndrome, things like this, where the body has stopped being able to identify the difference between self and non-self and has started to produce antibodies against its own tissue. We had some expertise in that area, and one of the questions that had been on the minds of folks for a long time is whether those self-directed, self-targeted antibody responses would occur in the case of a severe viral infection. We had good reason to believe that COVID was a potential candidate for something like that, and we thought that it was important that if that was happening to patients, that somebody investigated. So we did. We spent the next couple months. I would come in at about 8 o'clock in the evening. After some of my colleagues would have received blood from the ICU. We had a colleague that was consenting patients on the ICU floor, or rather consenting their families, because the patients were dying in the ICU on ventilators, so they couldn't respond, so we had to consent their families. So I would work with him. We would identify patients for study. Those patients' bloods would be collected by the local ICU nursing staffs. They'd be handed off to our own lab members. Those lab members would bring the samples back to the lab, and a lot of this was infrastructure built by An-Hyung Lee, who is also a faculty member here at Emory. We'd bring those patient samples back into the lab. They would be processed, and when I say processed, what I mean is the blood would be broken down into components so that we could study the cells in the blood alongside the antibodies in the blood off to the side. We were very interested in the cells because the cells are where the antibodies come from. So I would come in at about 10 o'clock, 8, 10 o'clock in the evening. I would pick up the cells from the most recent day's blood collection. We would go through a procedure where I would basically stain these cells. It's called flow cytometry. We won't get into it here. And then I would bring them over to a machine to analyze that blood. That would take me until about 12 o'clock, 1 o'clock in the morning. We would get the results that night. I would go home, take off all of my clothes, take a shower because it was not clear yet, again, if the virus could be transmitted from blood to, for example, my children, who at the time were very young, still are very young, had 2 kids at the time. One of them was under 1. So we were concerned that potential contamination could find its way into the household and that could make somebody seriously ill. So I would come home 12 o'clock, 1 o'clock in the morning, get undressed, take a shower, scrub down, get into bed, go to sleep, wake up at 6 o'clock in the morning, get the kids ready. We were fortunate enough to be able to have somebody come to the house to watch them so that I could go back into the lab. My wife would go back into the clinic where she's a physician. I could go back into the lab, start analyzing the data from the night before, start planning the next day's experiments, sit down with folks in the lab and spend the day essentially planning for the coming days and weeks. We ran at that pace, probably 17, 18-hour clips for, I would say, 2 months or so before it was very clear that we were seeing a very significant signature in the bloods of those patients. And that signature that we saw was surprisingly like the signatures that we saw in patients with acute, severe autoimmune flares. So if you took a lupus patient with flaring disease, so this is a patient with antibodies against their own DNA, against their own kidneys, against all kinds of things, experiencing really serious systemic disease. If you looked at the bloods of those patients, those bloods looked very similar to the bloods that we saw in patients with severe COVID-19. And in some cases, these were patients that were otherwise extraordinarily healthy. We had 34-year-olds males, for example, that were in the ICU dying of COVID at the time, who looked very, very similar to an advanced rheumatic disease or autoimmune disease patient. So by end April, early May of 2020, we had put our initial findings into a manuscript. We submitted that to an online repository called medRxives, so people could start to see what we were seeing in the bloods of these patients. We also started to ask the question, which was reasonable at the time, are these patients actually developing autoantibodies? So we went to the ICU, or a colleague of mine, the same guy that was consenting the patients on the ICU floor, went to his ICU colleagues and basically told them that it was time to start testing autoimmune patients for COVID-19 patients for autoimmune antibodies, right? The anti-double-stranded DNA antibodies that I was talking about and lots of other flavors of autoantibodies. He was able to convince them of that because those physicians were looking for ways to keep their patients alive. There was a standard of care, but the standard of care wasn't working. People were still dying, and so it was clear to people that we needed to find ways to track these patients beyond sort of our normal infection tracking protocols. So they started doing all of that, and we started to learn pretty quickly that not only did the bloods of these patients look like they might be autoimmune patients, but they were actually developing autoreactive antibodies. So these were antibodies against their own bodies. And that was happening within weeks of infection from COVID-19, seemed to be related to the disease severity. About that time, we published our first article. This would have been around August, September, October of 2020, so less than 6, 7, 8 months after we started the work. We published our first paper on the immune profiles of patients infected with severe COVID-19. That work has been cited now something like 800 times out in the literature. We were the first to talk in a detailed way. There were other papers, but in a detailed way about the development of these responses. So we kept on that track. Around that time, the New York Times was very interested in what was going on in these patients. We had an article published talking about the potential for autoantibodies persisting in patients. At the moment, or at the time, long COVID was just starting to be understood, really. So we had an article published highlighting some of the work that we were doing, talking about how some of these autoantibodies might persist in patients with complicated COVID-19 recoveries, and talking about some of the treatment strategies, hopefully, that might be available to them in the future. So all of that served as a foundation for about 3 or 4 years worth of work, where we really detailed the nature of the autoreactive breaks that these patients were undergoing, documenting its relationship to other infectious diseases. It turns out that it's not just COVID-19 that this is relevant to. It seems to be relevant to a number of other severe infections, at least bacterial pneumonia. If you get a bacterial pneumonia patient as serious as the COVID-19 patients that we were studying, you get a very similar self-directed antibody profile. All of those things got published. We moved on to long COVID. Long COVID, we found similar things, sort of the persistence of autoreactive antibodies in those patients. And we were able to start stratifying those recovery patients into functional groups where we felt like the potential for immunosuppressive intervention might actually benefit a discrete subset of patients. All the way through that, I had emails coming in from actual COVID, long COVID patients asking where the data was, what the data was looking like, how do we get them back to their normal functional selves. We had patients that had gone to, and these are all private communications that I've had, patients that essentially lost their lives, not in the terminal sense, but in the more subjective, I can no longer do any of the things that I used to be able to do, and I've developed sort of these symptoms that are so severe that there's not much hope that I'm going to get back to what I was. So we've been doing that now for 3 or 4 years. Along that way, the work was significant enough where I could make the case to develop my own lab here at Emory. We are a lab that is based on the idea that we have a really good detailed understanding of B cells, of the cells that produce antibodies, and that we can study those cells in lots of different contexts to benefit human health. That includes vaccination. It includes in infectious disease inside and outside of COVID-19. And all that is to say that yesterday we learned that the grant funding that encompassed all of the work that I just mentioned to you, so every bit of that work was cut. So we will no longer have access to those funds. I'm no longer the most direct recipient, so I will not be the person that is most directly harmed by that decision. But just for context, that grant was initially conceived of by the Trump administration, by the way, through the National Cancer Institute, to understand how patients with chronic diseases such as lupus, such as cancer, patients on immunosuppressive therapy, how those patients are responding to vaccines, not responding to vaccines, and how their disease state impacts our ability to protect them as an immunologic community. So we're closing the chapter on that. No longer relevant. I'm going to leave it at that today. Hope y'all are well.